Welcome to the third and final episode about our efforts to use vaults as vaccines. In my previous lesson, I described what happened when we vaccinated mice with vaults containing a foreign chlamydia protein. These foreign proteins are called antigens. We discovered that those mice were protected from infection, even though we didn't add an adjuvant. Remember, an adjuvant is usually needed to get the attention of the immune system, often by causing an inflammation. Since vaults containing an antigen were doing this themselves, it was a result that was as exciting as it was unexpected. When I presented those results to my colleagues, I wanted to be careful not to oversell them. The data were preliminary, and we couldn't assume that they would translate to humans. So, to keep things in perspective, I showed them this cartoon and said, "Good news, if you're a female mouse, we can protect you from chlamydia." Hi, I'm Lenny Rome, the Vault Guy. I'm a cell biologist and a nanotechnologist. My passion is a common particle of the human cell called a vault. With the help of my daughter-in-law, Tara, I am trying to explain what vaults are to non-scientists. Today, I want to cover three things. First, how we use a model antigen called ovalbumin to study the vault vaccine system. Second, I'll describe what this model revealed about how vaults interact with the immune system. And finally, I'll discuss why these results suggest vaults could become a new type of vaccine platform. Stick around and find out what happened when we vaccinated mice with a vault packaged with a protein isolated from eggs. The chlamydia results I described in episode 25 told us that vaults packaged with an antigen were doing something special with the immune system. They stimulated both antibody and T-cell responses without an added adjuvant. This is exactly what we want from a vaccine. Because we didn't understand how this was happening, we decided to back up and carry out some basic immunology experiments using a well-established mouse model and a standard antigen called ovalbumin, or OVA. Ovalbumin is the main protein in egg whites. Immunologists love using OVA because it is safe, well characterized, and most importantly, we understand in great detail how OVA interacts with the immune system of the mouse. As before, we produced a vault containing OVA using the INT targeting strategy, which I have described in several of my earlier lessons. Essentially, we make a fusion protein between OVA and the INT domain, which serves as a zip code to package the OVA inside the vault. Let's take a quick detour into how antigens are presented to immune cells. When a pathogen enters the body, it can be eaten by an antigen-presenting cell, or APC. The major APCs are dendritic cells, macrophages, and B cells. APCs act as the immune system's scouts. They capture foreign material, process it, and show fragments of it to other immune cells to kickstart the adaptive immune response. This process of eating the pathogen is called phagocytosis, something we discussed way back in episode 16. Here's a very fast review. The pathogen here is a bacteria. APCs take up the bacteria into an internal compartment called a phagocytic vesicle. These vesicles can fuse with a lysosome, and the bacteria can be chopped up and its proteins digested into small peptides. These peptides have sequences of amino acids that are not seen in human proteins. I don't have time to go into the interesting cell biology of how these peptides get routed to the APC membrane, but here is a simplified summary. The foreign peptides from the chopped up pathogen get transported to the cell's surface, bound to membrane proteins called MHC, short for major histocompatibility complex. Now, here is where it gets really interesting. T cells, the key players in immunity, are naturally attracted to APCs, and they can sniff out APCs that are displaying foreign peptides on their surface bound to MHC proteins. There are two classes of T cells: CD8 T cells, which are the cytotoxic or killer T cells that destroy infected cells, and CD4 T cells, the helper T cells that stimulate B cells to make antibodies and coordinate the overall immune response. These CD4 T cells play a critical role in protective immunity during a chlamydia infection. The CD8 T cells bind to one type of MHC, triggering cellular immunity, and the CD4s bind to a different type of MHC, triggering humoral immunity. A really effective vaccine activates both pathways, cellular and humoral immunity. Now, let's discuss the OVA-VALT experiment. We immunized 3 groups of mice. The first group was immunized with OVA alone as a negative control. The second with OVA plus a conventional adjuvant as a positive control. For this experiment, we used liposomes as the adjuvant. The third group was immunized with OVA-VALTs. 2 weeks after vaccination, we measured both antibody production and T cell activation. The results were impressive. Mice that received OVA alone made some anti-OVA antibodies, but did not have a strong cellular immune response. Mice receiving OVA plus adjuvant produced strong anti-OVA antibodies and a strong cellular immune response, both CD4 and CD8 T cells. Remember, an adjuvant is added to get the attention of the immune system, but adjuvants can cause problems like inflammation. What was most interesting was the immune response seen in the mice vaccinated with the OVA-VALTs. We saw production of antibodies and the stimulation of a robust T cell response, both CD4 and CD8 T cells. This type of cellular immune response is normally difficult to achieve without adjuvants or live pathogens. In other words, the VALT vaccine triggered both arms of the adaptive immune system on its own without an adjuvant that is usually required to attract immune cells. How could that happen? We got an important clue from a set of experiments that looked at which kinds of human immune cells in the blood take up VALTs. To do this, we prepared fluorescent VALTs by reacting them with a fluorescent dye. We then isolated all of the immune cells from blood and used a technique called flow cytometry to see which cell types internalized the VALTs. Flow cytometry lets us analyze hundreds of single cells per second by passing them through a laser beam and detecting the fluorescent signal. That tells us exactly which cells grab the labeled VALTs. What we found was striking. After just 10 minutes, about 23% of the total immune cells in blood took up the VALTs. The flow cytometry analysis indicated that of the cells that took up VALTs, 77% were dendritic cells, the main antigen-presenting cells of the blood. The other cells that took up VALTs were primary B cells, which are also antigen-presenting cells. In all, nearly 90% of the antigen-presenting cells in the blood internalized VALTs. These are the very cells responsible for starting an immune response. APCs in the blood are the immune system's gatekeepers. VALTs being preferentially taken up by these cells means the antigen is delivered directly to the cells that can announce it to the rest of the immune system. In short, VALTs appear to be trophic for antigen-presenting cells, meaning they are naturally drawn to and internalized by the immune system's frontline messengers. An empty vault that is eaten by an APC doesn't do anything. However, a vault containing an antigen engulfed by an APC results in the antigen getting chopped up and displayed on MHC receptors that are recognized by both CD4 and CD8 T cells. That allows simultaneous activation of CD4 helper T cells and CD8 cytotoxic T cells, explaining why we observed both antibody and cellular responses with the Vault vaccine without adding any adjuvant. It's as if the Vault particle carries its own delivery instructions, directing the antigen to the immune system. These findings suggest that vaults could form the basis of a new vaccine platform, one that's safe, stable, highly adaptable, and free of an added adjuvant. By loading different antigens into vaults, we could, in principle, design vaccines for a wide range of targets, from infectious pathogens to cancer. And because new antigen packaged vaults can be manufactured rapidly using yeast, a vault vaccine could be a viable substitute for mRNA vaccines, which require an adjuvant. Of course, there's still a lot to learn, but the idea that a naturally occurring human particle can serve as its own delivery system for immunity, that's pretty exciting. Maybe one of these days, I'll be able to swap out that cartoon of Minnie Mouse with an actual person. I'm Lenny Rome, the Vault Guy. Normally, this is where I tease what's coming in my next lesson, but it's still under construction. So, like much of the world of vaults, it will remain a mystery for now. In the meantime, please remember to like and subscribe.